Good evening all of you. Today in our botany session, there is very important topic from the chapter Biomolecules, that is nucleic acids. This topic is linking two chapters. One chapter from your +1, which you call it as Biomolecules. Second chapter is from your +2 NCERT book, what is molecular basis of inheritance. Their introductory part, first 2-3 pages I must say, is linked to this chapter. Fine. So indirectly we are coming with the two topics from two different chapters. Right now I am considering it under the biomolecules. Let's start with this important topic. What is the topic right now? Nucleic acids. This nucleic acid that was discovered by the scientist, this is very important, you can say one of the macromolecule. I must say this word to you to get into its history. So its discovery was done by the great scientist Friedrich Mischer. But he named it as nuclein, not as a nucleic acid. The term nucleic acid that is coined by that was the scientist Altman. So this is small regarding its history regarding nucleic acid. Who discovered that? Friedrich Mischer, but he named it as nuclein. But who had given the term nucleic acid? That was by the scientist Altman. And in between I introduced one word to you, that are macromolecules. Means they are having molecular weight more than 10,000 Daltons. Let's see what are the types of this nucleic acid. If I am taking for the types of this nucleic acid, it will be basically of two types. What is that? DNA and RNA. And keep in your mind, the nucleic acid will be the genetic material in the living organism. This is very important to understand. DNA in all living organism will be the genetic material. But if I am saying in all the organism according to your NCERT book, then I must say that in some viruses it will be the RNA as a genetic material, right? Not in all the organism, being viruses, some viruses are having RNA as genetic material, but all living organisms, they are having the genetic material as DNA. That organism where DNA is genetic material, do they have RNA? Yes, such organism will have RNA. Then what is the function of RNA in such type of cells? This RNA will be working as an messenger that can be acted as an adapter, that can be help in the formation of structure, and even it can have the catalytic role. What is an adapter? How it is sending the messages? This we will deal when we will take in our classes the topic, the complete regarding molecular basis of inheritance. There we will discuss about transcription, in which messenger RNA will form. Adapter RNA is the tRNA, which is translating the language of nucleic acid into the proteins. And as a structure, you know that it helps in the production of the ribosomes. Ribosomes are the protein factories of the cell. And catalytic role again by rRNA, not all types. For example, if I am taking 23S ribosomal RNA, ribosomal RNA have different sedimentation coefficient based on that different types of rRNA. Which rRNA will be acting as a catalyst, means as an enzyme, that is 23S ribosomal RNA, but not all the enzymes are going to be ribozymes. Almost all enzymes are protein. Almost all means few are what? Made up of this RNA nucleic acid. Now to coming back to our main topic, if I focus, let's recapitulate this point. Can I say in all living organism, what can be the genetic material? DNA, which you call it as deoxy ribonucleic acid. Right? In such organism, do they have RNA? Yes, they have RNA. Then what is the role of RNA? If it is not, is a genetic material. This can act as messenger, this can act as an adapter, can act as a structure forming, and even catalytic role it can play. But viruses which are having RNA as genetic material, they cannot have DNA. Or if viruses are having DNA as genetic material, they cannot have RNA, right? So this basic thing must be there in your mind for the introduction of this topic. After this, let's see what are the constituents of this nucleic acid. Can I say this is a polymer, right? Which type of polymer? It is an heteropolymer. Heteropolymer means their subunits are different types. That's why you call it as heteropolymer, and polymer is what? Macromolecule. If I am taking this for the nucleic acid, then obviously what monomer it is made up of? Every polymer, poly means many. If many small units link, that you call it as bigger thing, which you call it as polymer. And here that polymer name is nucleic acid, and obviously a polymer is made up of the monomers. What monomer is there in nucleic acid? I am taking either of DNA or RNA. What can be its monomer? That you call it as nucleotide. This is its monomer. But this nucleotide is further consists of their components. What is that? Like it is made up of pentose sugar. Along with that, it is made up of nitrogen base. Even it is made up of phosphate group, or you can call it as phosphoric acid. To discuss nucleic acid, first of all you should have good knowledge about their monomer. What is that monomer I have taken? Nucleic acid. And what are its component of one nucleotide? It should have pentose sugar with nitrogen base and with one phosphate group. Now if this pentose and nitrogen base combine, that you call it as nucleoside. Can I say nucleoside plus this phosphate group? Can I call it as phosphate group? Together they are forming one nucleotide. Don't say that nucleoside is a monomer. This is very important. Sometimes students are making mistake and saying that nucleoside is a monomer of nucleic acid. It is not like that. Nucleoside, when bonded with the phosphate group, with the phosphoric acid, then it is forming one monomer of that, which you call it as nucleotide. Now let's take all the components one by one. I am taking first of all phosphate group. In that, for the phosphoric acid, what is the formula of this? Molecular formula H3PO4. And what is the structural formula for this? Like this, we can represent it structural formula. This is for one component among the three. Next component, I am taking pentose sugar. This pentose sugar, as the name is indicating, is having 5 carbon, but closed ring it will be, which is heterocyclic. If I am taking DNA and RNA, What DNA is carrying which type of pentose sugar? Deoxy ribose sugar is there in DNA, right? Then in RNA, what is that? Ribose sugar. Let's see how it is heterocyclic, how it is pentose, and how the carbons are going to be arranged. I am taking for ribose, it is having oxygen in its ring, and these angles are indicating for carbons. If you will count the number of these angles, these are 4 in number, but just now I said to you it is pentose. Then where is a 5th carbon? It is in a side branch like CH2 having the OH group. Now let's complete for each and every carbon. If I am taking this as 1 prime position, 2 prime, 3 prime, 4 prime, and this is 5 prime. What is there on 1 prime? On the 1 prime, the OH group is on upper side and H is on lower. And on 1 prime, it is having H, OH on lower side. Here H and OH, here will be H. I already mentioned, upside is C2OH. Now, what is happening? Can I say RNA is having such type of ribose sugar, which is having how many carbons in the ring? In the ring, it is having 4 carbons. Then where is a 5th carbon? This is in a side branch. Fine. Now, how we can say this is pentose and this deoxyribose is a pentose but derived from ribose sugar. How it is that? See its name. The word is what? D. D means without. Oxy means oxygen. Somewhere oxygen is not present. Let's see at what position. If I am taking this structure and keeping it like this, same like that, ribose I am drawing at 1 prime position OHH, here will be H, but on this 2 prime position you will find it is not having OH, it is having hydrogen. Rest all positions will have their respective H and OH. Where there is a loss of oxy group means oxygen at 2 prime position. That's why sugar present in DNA you call it as deoxyribose sugar. We take it deoxyribose sugar is derived from the ribose sugar. Why we call it as heterocyclic? Because in a ring all are not carbon. One is oxygen, rest are the carbons over there. This is about the second member in the nucleotide we have discussed. Let's see for the nucleotide. Means this is about the nucleoside. Now I am taking after this for nitrogen base. Next member in nucleotide. Nitrogen bases are grouped or categorized under 2 categories, purines and pyrimidines. Right? Purines and pyrimidines both are heterocyclic. Hetero means all members are not same in the ring, but not due to oxygen, it will be due to nitrogen now. That's why you call it as nitrogen bases. Which 2 purines are there in nucleic acid? That will be adenine and guanine. And what pyrimidines are there? Uracil will be in case of RNA, thymine in case of DNA, whereas cytosine is in case of both DNA and RNA. Can I say cytosine is such type of pyrimidine which is common to both RNA and DNA? But what is not there in DNA? Uracil. That pyrimidine is not there in DNA. And which pyrimidine is not there in RNA? Thymine. Fine. But purines are present, adenine and guanine, both in DNA as well as RNA. Now let's see the structures of purines and pyrimidines one after the another. In that process of discussion of this nitrogen bases, I will take first of all regarding purines. first I am taking for adenine. It is having the structure like this. One ring, and being this is dicyclic, you will find here there is a presence of 2 cyclic ring, not 1, and it will be coming out like this, right? I repeat, keep in your mind, purines, pyrimidines are heterocyclic. What is the difference basically? Types, number 1 we have already discussed. Now I am telling you purines are what? Dicyclic. When I will discuss with you pyrimidine, they are monocyclic, means it will have only 1 ring there. But in both the cases, purine and pyrimidine, it will be heterocyclic. Now let me to draw it complete. So I am not taking valences completion, hydrogens I am not taking. So nitrogen of valency 3, carbon of 4, you know very well. Simply I am making the bonds right now. Here will be NH2 group. This is the structure of adenine. Can you see dicyclic? And both the rings that is having carbon as well as nitrogens. That's why you call it as heterocyclic. Now here somewhere numbering is important. Why I need to say numbering here? Because when we will discuss about its bonding, at that time you need the numbering. So how you have to number this? In this ring you have to go anti-clockwise. From this is first position, second, third, fourth, 5th and 6th. Here will be 7, 8 and 9. This you have to go clockwise in writing, here you have to go anti-clockwise. Now this numbering, number 1 is indicating what is the position of nitrogens here. Can I say nitrogen position here will be 1, 3, 7 and 9. This is for adenine. And at which position amino group is attached? At 6th position. Now let's see for next purine. What is that? We have taken in that aspect 2 categories, purines and pyrimidines, right? I am taking that slide back again, this one. Okay. So here we are taking for guanine. Now what is happening in its structure? Carbon, 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 nitrogen, carbon and nitrogen. Can you see heterocyclic? But it is dicyclic as well. And we are completing bonding here right now. But it is having here oxy group and here it is having amino group. Now what is the numbering trend here? I already spoke to you. It is having 1, 2, 3, 4, 5 and 6. Here will be 7, 8 and 9. Position of nitrogen is fixed for purine. What is that? 1, 3, 7 and 9. Both in adenine and guanine. Fine. Now what is the difference between the adenine and guanine? Can you see 6th position oxy group is there. Now amino group is on the 2nd position. This is the difference between them. Now after this coming to the pyrimidines. For the pyrimidines, I said you it is monocyclic. The first one I am taking for uracil. Now what is there in a uracil? Only 1 ring will be there, which is heterocyclic and having oxy group at 2 position. Now how to number this? We have to number it clockwise. How we have to number it clockwise? Starting from this nitrogen, 1, 2, 3, 4, 5 and 6. Now can you see what is the position of nitrogen here? The position of nitrogen is 1 and 3 in uracil. But oxy group will be there at position 2 and 4. This structure is for uracil, and uracil you know that nitrogen base in case of RNA, not in case of DNA. Now coming to the next structure, which is of thymine. There is what? Carbon. It is almost same like we have discussed for the uracil. Only one difference is there, that on 2nd carbon, no doubt here will be oxy group. But when you see the 5th carbon, 1, 2, 3, 4, 5, 6. On the 5th carbon, there is a presence of methyl group. I can call thymine as 5-methyl uracil. What can I call it as? 5-methyl uracil. So it never means 5-methyl uracil is a uracil. No, 5-methyl uracil is thymine. Keep that in your mind. This is very important thing that you have to go with. Fine. Now after this uracil and thymine, we have to discuss the third pyrimidine. What is that? Cytosine. Now what is the structure of cytosine? It will go like this, monocyclic. Now what it is having? Amino group here, but here will be the oxy group. Now how it have been having the numbering? Like same 1, 2, 3, 4, 5 and 6. And here they are having double bond here is there, but here will be double bond here as well as here. So keep in your mind, these three structures are for what? Pyrimidine. The main motive of drawing this structure is to get familiar that they are heterocyclic. Monocyclic if I am saying, then it will be pyrimidine. But if I want to say that it is heterocyclic as well as dicyclic, that means I am discussing about what thing? Purines. That means I am discussing about the purines at that point. So all the structures are right now in front of you. 2 for the purines and 3 for the pyrimidines. Means 3 different structure for pyrimidines. Keep in your mind, for both the purines, the position of nitrogen is 1, 3, 7, 9. And 3 pyrimidines, uracil in RNA, thymine in DNA, cytosine both in RNA and DNA, the position of nitrogen is 1 and 3. Next thing what you have to take care of, when you are looking to all the structures, can you see in which nitrogen base oxy group is absent? That is in case of the adenine. The oxy group is absent, it is only having amino. Rest all either 1 or 2 oxy group are there, or 1 in the last. In cytosine, they have oxy group. But complete oxy group is absent in case of adenine. So this is about the structures of the nitrogen bases we have discussed. So can I say we have discussed about all the 3 components of what thing? One nucleotide. Now let's see how these 3 are combined now to make one nucleotide. Means we are telling you about now the bonding which is present to create one nucleotide. For this thing, let's draw for the structure of sugar first of all. Which sugar? Deoxyribose sugar I am taking in context to the nucleotide. Which nucleotide I am drawing? Deoxy nucleotide. The word deoxy is indicating this is a nucleotide of DNA. Fine. So this is the pentose sugar which is deoxyribose. Keep in your mind, prime position are for sugars. If I am not writing prime, simply 1, 2, 3, 4, 5 like this, it is for position in the nitrogen bases. So for the deoxyribose sugar, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime will be on side branch, CH2OH. This will be 5 prime. I said you in this pentose sugar where the base is going to be attached. The base is going to be attached to make it what thing? To make it nucleoside. At 1 prime position, the base is going to be attached. And the mind should be on the structures now. If that base is purine, then where this bond is going to create? The bond is going to create on which nitrogen? Then it will be on 9th nitrogen. Right? Let me to draw it for you. If it is purine, I am taking for purine now. What is the structure of purine? like carbon, 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 nitrogen, here carbon, nitrogen, amino group I am taking means adenine I have taken, here is nitrogen, carbon, nitrogen, here is the hydrogen, double bonds will be here, and at this point 1 prime, I already said you upside is OH and here will be H. What is going to happen now? There is a loss of water molecule. When this water molecule is going to be eliminated, there is creation of bond. This bond you call it as glycosidic bond. And in this bond, carbon is making bond with nitrogen. That's why you call it as N-glycosidic bond. So can I say glycosidic bond is created by loss of water? Fine. So can I say this is a nucleoside in front of you. To make it nucleotide, I have to attach here phosphate. Now where it is going to be attached? I already mentioned you the structure of this is like this. Fine. It is making bond with the COH means CH2OH which is on 5 prime. Again here, let me to make it clear, there is the loss of water molecule. If water molecule is going to be lost, can I say now there will be creation of a bond which you call it as phosphoester bond, right? So where the phosphoester bond is created at 5 prime position and where the glycosidic bond is created at 1 prime position. This is one deoxy nucleotide in front of you. How can I say that? Because here instead of OH, H is present. On that grounds I can say this is a nucleotide of a DNA. Fine. Now this is the bonding to create one nucleotide. Now whatever base is there, here I have taken for purine, it is binded at 9th position. If it is pyrimidine, then the bonding will be at nitrogen number 1, right? If it is a purine, then bonding at 9th nitrogen, and if it is pyrimidine, then where it is binded, it is going to bind here. It is having hydrogen here to complete the valency of nitrogen of 3. So again water molecule will be eliminated and you will get there the purine attached with the sugar. Purine is attached to the sugar at 9th nitrogen. Pyrimidine is attached to the first carbon here with the first nitrogen over there, and both are forming glycosidic bond. But at a time either purine will be there or pyrimidine will be there, right? Let's repeat this point once again. Pyrimidine if it is there, it is binding to first prime carbon at which position? First nitrogen. If purine is there, it is bonded by 1 prime position at which nitrogen? 9th nitrogen. Fine. So this is the structure of one nucleotide. Now let's see what nomenclature we have to use for all these things. What is that? If I am taking the heading, which nitrogen base is there? If nitrogen base attached to the sugar, then what name is given to the nucleoside? And if nucleoside is attached to phosphate, then what name is given to its nucleotide? Let's see for purines first of all. If I am taking purine here, adenine, then what is their nucleoside name? Their nucleoside name will be adenosine. If I am writing simply adenosine, that means this is nucleoside of RNA. If I am keeping deoxy word here, deoxyadenosine, then it will be of DNA. Are you getting that? So here simply I am writing for the RNA to avoid the confusion. So here adenosine, when phosphate is attached, then you call it as what thing? Adenosine monophosphate. Fine. Or even you can call it as adenylic acid. right? Being phosphate group have been attached, which is acidic in nature. Phosphate group is providing acidic nature to the nucleic acid. I repeat, if I am writing all the words without deoxy, I mean to say this is for RNA. If I am keeping deoxy word, that means these all terms are for whom? DNA. And if I am writing acid, this is for RNA. Now next is what thing? Guanine. For guanine, what is the name of the nucleoside? Guanosine. What will be the name of its nucleotide now? For guanosine, it should be guanosine monophosphate, or even you can call it as guanylic acid. So this is about purine. Let's see for pyrimidine now. If I am taking base pair pyrimidine, first I am taking uracil. Here the nucleoside will be uridine. Keep in your mind, in uridine you need not to write deoxy because it is understood it is only in RNA, not in case of DNA. Same for thymine and its thymidine. You need not to write deoxy because that is understood it is there in DNA, not in RNA. So uridine, I repeat, it is in RNA, not in DNA. Now coming to its nucleotide, it is uridine monophosphate, or even you call it as uridylic acid. Fine. Now what is the another nitrogen base which is pyrimidine? Thymine, which is in DNA, not in RNA. It will be thymidine, and what you call it as nucleotide? Thymidine monophosphate, or even you can call it as thymidyllic acid. Last we are left with the cytosine. Cytosine, you call it as cytidine when it is forming the nucleoside. You are taking deoxycytidine if I am talking about DNA. Simply writing this means RNA. Then what we can call it nucleotide as? Cytidine monophosphate, or even you can call it as cytidyllic acid. Right? Why I can say that this part is important? Reason many questions can be framed. For example, if I am saying you what is the difference between guanosine and guanine, then you should know guanosine is made by sugar and the nitrogen base, whereas guanine is only having itself as a nitrogen base. If I am discussing guanosine monophosphate, what is its difference from the guanosine? It is having phosphate and it is not having phosphate. If you know the terms, only then you can handle such type of question. And here interesting thing to know is that when you see the nucleoside of the purines, can you see it is ending with the SINE? And definitely students get confused when they see this ending at SINE. What is cytosine? This is only nitrogen base. And what is adenosine and guanosine? These are nucleoside. So you should be very careful about terminology. Wherever you get some sound same, check it, is that same thing or different thing. Now one more thing, if I am writing cytosine, this is a nitrogen base. As an extra note, let me to write it for you as an extra note. What is that? If I am writing the word cysteine, this is not a nitrogen base. This is an amino acid, totally separate topic. This is totally separate topic from here. So looking to the spelling, don't get confused. Cytosine is an what? Nitrogen base. Adenosine, guanosine are what? Nucleoside. Nucleoside means having the sugar plus nitrogen base. And what is cysteine? This is an amino acid. It is not linked to our topic, but still I am telling you, if options are there, you should be very careful in choosing the option according to question what they are asking. Fine. So here what we have discussed, the different terms related to the nitrogen base, nucleoside and the nucleotides. Coming to our next important topic, which we have to discuss now in the form of nucleic acid, means now we have to assemble the nucleotides to create a long chain of polynucleotides, which you call it as polymer. Now if I want to make polynucleotide, then how it will be created? If I am taking again this as deoxyribose sugar, here I am saying CH2 and here phosphate is there. On this side I am taking base, right? Not going in much complex or full structure, simply I am saying base is there which is formed by glycosidic bond. And which bond is here? This bond is the phosphoester bond. And what is this bond? Glycosidic bond. Just now we have discussed. Now how this one nucleotide is going to bind with the adjoining nucleotide, even it is having its own phosphate group. These are two nucleotides in front of you. Let's see how they bind to make a polynucleotide chain. This thing can happen both in DNA or RNA. But RNA is single stranded, DNA is what? Double stranded in the living organisms. I am not talking about viruses now. So here the DNA is double stranded, RNA is single stranded. Let's take in one strand of both how bonding will happen between the nucleotides. As you already know that they are having phosphoester bond at which 5 position? I mean to say which carbon position? 5 prime position. Same for here. What is this bond? Phosphoester bond. What is this bond? Again phosphoester bond for this nucleotide. Now keep in your mind this phosphate is having one more OH. 3 OH is there. I already shown you. One OH is occupied here to make phosphoester bond. Second now participate in this OH of neighboring sugar. And again there will be loss of water molecule. If here this water molecule is going to be lost, then this carbon and oxygen, they are forming which bond? This you call it as phosphoester bond. Usually students are mistaking it up and saying it as phosphodiester. No, again it is what? Phosphoester bond between this phosphate and 3 prime position of neighboring nucleotide. I repeat this thing, please listen it very carefully. This nucleotide have its own phosphoester bond at which prime position? 5 prime. But this is forming the phosphoester bond at which prime position of this nucleotide? 3 prime position. Now can you imagine, this is phosphoester bond, I am writing in short now, this is phosphoester bond, even this is what bond? Phosphoester bond. Now this complete you call it as phosphodiester bond. What do you call it as? Phosphodiester bond. If I am taking this as phosphoester bond, so it is having one phosphate and it is free end and you call it as 5 prime end. Now let me to take one more, because it is a very long chain. I am taking few to explain this. Let's see one more. It is whether deoxy or deoxyribose sugar or ribose sugar. Anyway, it is going to make the polynucleotide chain in the same manner. Here is a base, glycosidic bond having CH2 and having its own phosphate. Which bond is there? Phosphoester bond of this nucleotide at which prime position? 5 prime position. Now this phosphate will make a bond with the neighboring nucleotide at which prime position? 3 prime position, and this bond even you call it as phosphoester bond. Now from here to here you are having 2 phosphoester bond, that's why you call it as phosphodiester bond. Are you getting this point? Likewise, we are getting long chain of nucleotide which you call it as polynucleotide. And can you see here the free end is 5 prime. Here the free end is what? Can you see? It is 3 prime end, right? If RNA, then it will be single stranded. If it is DNA, I have to draw the second strand which is anti-parallel. Anti-parallel means 3 prime on upper side, 5 prime now on lower side. Here 5 prime is upper, 3 prime lower. Now in other strand the 3 prime is upper side and 5 prime on lower side. How I have to draw? Now see here, we have to take it inversion of this like this. Now what is happening? This will be 1 prime, 2 prime, this is 3 prime, 4 prime, this is 5 prime, and here is its phosphate. One more I have to draw like this to make you clear. This is 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. At this it is having phosphate. What will be at 1 prime position? Base. What will be at 1 prime position? Base. Likewise, one more I am taking like this, this and like this. Here again 1 prime, 2 prime, 3 prime, 4 prime, 5 prime is having this phosphate and 1 prime is having the base. As per I already said you, they have their own phosphate as which prime position? 5 prime. What is this bond? Phosphoester bond. But it is making bond with the neighboring at which point? 3 prime position. And even this is what? Phosphoester bond. And together you call it as phosphodiester. Likewise, it is making bond here as well. Now how the bases are linked with the opposite? Can I say this is linked by hydrogen bond? If 2 hydrogen bonds are there, that means this base can be adenine thymine or thymine adenine. If they are having 3 hydrogen bond, then it will be cytosine and the guanine, right? And so on. If I am taking like this, maybe guanine or cytosine like this. So like this we are having 2 strands of DNA, or I can say 2 polynucleotide chains are there in DNA, then their ribose sugar is not there, you call it as deoxy ribose sugar. And can you see now free end is what here? 3 prime. Here is what? Free end here? 5 prime. So keep in your mind, in DNA 2 polynucleotide chain but anti-parallel to each other. One is from 5 prime to 3 prime, second is from 3 prime to 5 prime. Fine. So like this we are getting gradually the double stranded DNA. Now let's check about some of the salient feature of the DNA model or double helix model given by Watson and Crick. According to Watson and Crick, double helix model of DNA which is based on, one more thing you should know, this double helix model of DNA that is based on X-ray diffraction data given by the 2 famous scientists. What is that? Rosalind Franklin and Maurice Wilkins. These 2 scientists has given through X-ray diffraction data about the structure of the DNA. But Watson and Crick use this data to figure out the or to propose the structure of a DNA. But add on to that, they have taken the authenticity from the Chargaff's rule. What is the Chargaff's rule? Chargaff's rule, in short, I must say here right now, which is saying that in a DNA, purines, the amount of purines will be equal to the amount of pyrimidines. Means if I am taking adenine plus guanine, that must be equal to the cytosine plus thymine. Or can I say A plus G divided by, if I am taking on this side, C plus T, it should be equal to 1, right? Next thing, the amount of sugar molecule must be equal to phosphate. This is a Chargaff's rule. The authenticity is even based on the Chargaff's rule for the double helix structure of the Watson and Crick, what they have given for DNA. Let's check now its salient feature. To figure out for its salient feature, I am taking the double helix structure of a DNA like this. Roughly I am drawing that and eradicating this part out. Now when you will see such structure, you will find it is having two grooves. This is minor groove, this is a major groove. Can you see it is deep? It is shallow. This shallow groove you call it as minor groove, and this one you call it as major groove. One minor, one major, together you call it as one helix of DNA, one turn. You call it as one turn of DNA. Right now we are discussing which DNA? B DNA. Because based on the number of nitrogen bases per turn and other many more factors, we are having different types of DNA. Right now you should know we are discussing the detail about B DNA, which is having 10 base pairs per turn, right? Like this is one nucleotide chain, polynucleotide chain, this is second. They are linked by what thing? They are linked by hydrogen bonds. Can I say, suppose here adenine, here is thymine, this is one base pair. If it is cytosine guanine, this is second base pair. Likewise, in one complete turn or helix, how many base pairs will be there? 10 base pair if I am taking the B DNA. Now, can I say this structure is having two polynucleotide chains? Polynucleotide chains whose backbone is made up of sugar and the phosphate. Now what do you mean by backbone? If I show you the previous structure, in the previous structure of DNA, can you see this is phosphorus, sugar, phosphate group, sugar, phosphate, sugar. This is a backbone for both the polynucleotide chain. Where are its bases? Bases are projected at right angle to that, but towards inner side. They are not towards outer side, they are towards inner side, but right angle to this backbone. Fine. So this is the salient feature what they have said for the double stranded DNA, that two polynucleotide chains will be there and whose backbone is made up of sugar and phosphate group. Now can I say where are the bases? Bases are projected at the right angle towards inner side, and if adenine is there, what should be opposite? Thymine, bonded by 2 hydrogen bond. If cytosine is there, it is bonded by 3 hydrogen bond. Cytosine with guanine by how many hydrogen bond? 3 hydrogen bond. Now its pitch of helix or helical pitch is having the measurement, means the one turn measurement. If I am taking vertically, it will be going to be 34 angstrom, or you call it as 3.4 nanometer, right? Now what do you mean by this thing? If I am taking this as one turn, its vertical distance, this length of one turn will be 34 angstrom or 3.4 nanometer. And in that turn, how many base pairs will be there? 10 base pairs will be there if I am talking about bDNA. Fine. Okay. Now what is the next point? Can I say one base pair is stacked upon another, one above the other? Now see here all of you, if I am taking here one base pair like this, second will stack on that but not like this at angle of 36. Now next is stacking on that on 36, next 36. So every base pair is piling upon each other at what angle? 36 degree. Total how many base pairs are there? 10 base pairs are there. So one turn will be completed by 36 multiplied by 36 degree multiplied by 10. What degrees? 36 degree will be one turn is going to make a complete helix. It is going to turn completely in one turn how much? 360 degrees. And this hydrogen bonding is additional thing which is giving stability to this structure. And this structure is what structure? Secondary structure. And this is right-handed coiled structure. This is right-handed coiled structure, means it is going to be turned towards the right side. We are having exceptionally one more DNA which you call it as zDNA. It is the only DNA which is left-handed coiled. Right? Rest all the types of DNA like in our nature A, B, C, D and E. Just now I said you Z even. These are the types of DNA. These all are right-handed coiled. These all are right-handed coiled DNA. But this one only is what just now I said you, left-handed coiled DNA. This is having base pairs per turn. How many here? 11. It is having 10, just now I discussed with you. Then is for 9, 8. This is having 7.5. Means 8th is shared between the two turns. That's why you are calling it as here 7.5. Z is having how many base pairs per turn? It is having 12 base pairs per turn. Fine. But we are discussing about which DNA? bDNA. Fine. So what we have discussed, that is about the structure of our bDNA, which is right-handed coil and having the polarity anti to each other. Anti means if this is 5 prime ending into 3 prime, this must be 3 prime to 5 prime. Can I call them that they are anti-parallel to each other? Where the backbone is made up of what thing? The backbone is made up of sugar and phosphate. Where the bases are there? They are towards the inner side, right angle to the backbone. Fine. So these are the basic point for the salient feature of the main nucleic acid, which you call it as master chemical of the cell. What is a master chemical of the cell? DNA. In living organism, who is acting as a genetic material? DNA. Is the RNA is there? It is there, but it is having other function that we have discussed in the beginning. So this is all about an important topic. What is that? Nucleic acid. I hope so you have learned a lot in this. Do revise that, and this small portion will link to your chapter. What is that? Molecular basis of inheritance in the introductory part as well. This is all for today. Thanks a lot, students.